All right, so I can begin. I haven't wrote the exam quite yet. I've started it. I can give you, students always like to know this. I'm not sure why, but if it helps, it helps. There will be about 32 questions on the exam. It'll be roughly speaking 25 multiple choice questions, 4 short answer questions, and 3 matching questions, something in that ballpark. But I would expect somewhere between 30 and 35 overall questions, with the majority of them being multiple choice questions. Okay, and now what I'd like to do is just take questions from all of you. You can either speak the questions or you can put them in the chat. I can see some, okay. Will it have a kind of even split of questions between each chapter? When I wouldn't say from each chapter, but I would say from each day, which is roughly each chapter, but on some days we covered certain chapters differently. When I make the exam, I go through the note-taking guides and I try to have questions from each learning objective from each note-taking guide. And usually it's more than one question from each one to spread it out. So if you're studying the note-taking guides, you should have you should be able to see, you will have, let me back up, I'm sorry. If you're studying the note-taking guides, then make sure you understand each objective, because there'll be questions from each objective. So I think that's the best way to study for it. Look at the note-taking guides. And if you haven't completed them yet, that's okay. You can still look at them. And if you can complete them based on the notes you've taken and what you've learned in class and the homework and all that fun stuff, then you're in pretty good shape. If you've completed the note-taking guides, then you can just review those, or maybe open up a blank one and see if you can answer the questions. I think that's a good exercise. Don't forget to look at the application questions. Every single year, there's a few students that don't do well because they didn't study the application questions. Make sure that's not you. Make sure you look at the application questions. On Blackboard, you will see all the application questions we talked about and the take-home points after each one of those. And there's a few extra application questions that we didn't get to in class, and you can look those over as well, just for extra stuff. All right. Jessica, you asked, can you go over the application question about the microscopic worm? Oh, do you mean from day one, I think, right? I can, let me, I'll just pull it up. There's not much you really need to know from that, but let me tell you what you do need to know. If someone can't see this PowerPoint, speak up and say something. Don't just put in the chat, verbally say, because I might not be able to see the chat when I'm looking at the PowerPoint. But you should be able to see this. And on day one, we talked about the Hopi Native Americans, and then we had this one. We didn't do it in class. And for this one, what I really want you to know is the importance of model organisms. Briefly, I can tell you what we did, but I'm not going to test you over this. Essentially, we looked at some videos showing worms and how they normally behave. We added alcohol to them to see how they behaved with alcohol. And then we looked at mutations that reversed that effect. And then if we talked about this class, then we would have talked about how is that applicable to humans. But the main take home I want you to get from this, and the thing that could appear on your exam, is that model organisms like C. elegans here are good organisms to study to learn more about humans, because of the evolutionary link between humans and C. elegans. So studying this simple organism tells us a lot about us as humans. That's the part I want you to take from this. And if you have any other questions, Jessica or somebody else, just you can put them in the chat, or you can just say them out loud. But you should know that they are good representatives to understand humans. Yeah, which cells do we want telomerase in? Let's go to that. Let's find that application question here. Here it is. So telomerase is important in maintaining the proper length of chromosomes in cells. The size of oh, that's the take-home point. I'm sorry. Let me. Okay, not all cells in humans, I think this is the one you're talking about. Not all cells in a human have telomerase expressed. Which cell types would we want telomerase activity, and which cell types would we not want telomerase activity? So remember what let's go here. So we have a chromosome here. Just draw it like so. And the telomerase at the end here, after every time this cell divides, each of these, got rid of the whole line. Let me refer to that here. Every time it divides, the chromosome gets shorter. Okay? Every time it divides, it gets a little shorter. And the ends of the chromosomes have very few genes on them. So by cutting the ends short, you're not causing problems. But at some point on the chromosome, you will get to a part where there are genes. Sorry, that's a horrible arrow. But there are genes here. And once it gets to the part where you're starting to remove genetic material, that's where you start having some problems. And we don't want that. And usually about that time, the cell, the ends of the chromosomes also become a little sticky, a little less stable. And that triggers the cell to go under apoptosis. And we want these cells to undergo apoptosis, because if they don't, they accumulate mutations, and then those mutations can lead to cancer. So the types of cells we want to die at a regular interval are cells that we worry that they could get cancer. So like skin cells, cells that line, our GI tract, cells that divide a lot. Those are cells we want to have to die. And in order to have this telomere shortening, we cannot have telomerase expressed. No telomerase means the cells die at a regular pace. And that's what we want for cells that could accumulate mutations and cause problems. The only cell types that we want to have telomerase activity present in are stem cells, and really mainly just stem cells. But to define that a little bit more, I will. So stem cells. So this includes the adult stem cells that we currently have in our body, like our stem cells that are in our liver, our pancreas, our skin, our fat tissue. They all have stem cells, and they're important because those stem cells rejuvenate, make new copies of those specialized cells in our liver, our pancreas, or what have you. And so when those different cells die, they're replaced by stem cells. We want those stem cells to have telomeres that remain the same length. And the reason for that is that when that stem cell becomes a specialized pancreatic cell, say, we want it to start off its life as a specialized pancreatic cell with the proper length of the telomere or the proper length of the chromosome. If it's short because telomerase isn't active in these cells, and the stem cells make a specialized cell, it's not going to live very long. It's going to create problems. So you want stem cells to have proper lengths of chromosomes so that when they become specialized, they have a typical life of that specialized cell. So stem cells, we want telomerase to be expressed. Specialized cells, we don't want telomerase to be expressed. embryonic stem cells, same difference. We want all of our embryos to have the proper length of our chromosomes so that when the embryo becomes a formed human, right, and then ultimately at some point becomes an adult, we want them to have that full complement of telomeres. Paige, you asked that question. Does that help, or did you want me to expand on something? Or anybody else can also talk about this. And the bottom question is, why are cancer researchers interested in telomerase activity? Because it tells us something about the life of a cell. And we know that cancer cells often have telomerase expressed. And that's the reason cancer cells are immortal and they can keep dividing and surviving. It's because their telomeres don't get smaller. So in cancer cells, often the telomerase gene is incorrectly expressed, and that causes problems because those cells just don't regularly die. Any other questions? Any concept that's confusing or any activity you'd like me to go back over again? Any homework question? See, a couple people are typing something, so I'll wait. Quickly go over each of the studies. Well, I don't know if I can quickly go over each of them, but I can look at them. And if you can help me and tell me, when you say the studies, which studies do you mean? Do you mean the studies that showed that DNA was the genetic material? You want me to talk about Musil and Stahl? Do you want me to talk about all of them? Where you want me to focus? Okay. Semi-conservative and why it is significant. Okay, we can. I think what I'm going to do here is just randomly put in a new slide here. Okay, so semi-conservative replication. And remember, semi-conservative replication is what's actually happening in our cells. Every living organism uses semi-conservative replication. And in essence, what that means is that when you have a cell that has the chromosomes in it, right? We have one copy of every one of our chromosomes from the maternal source and the other from the paternal source. With semi-conservative replication, what that means is the final product after one round of replication, for half of the chromosome is conserved in each resulting cell. Okay, so remember, this is a cell here, and it's going to make two new cells. These are horrible pictures. I'm sorry about that. But when they make a new cell, and that process, they're going to split the chromosomes down the middle, And so each of the new chromosomes, one of them is going to have the red one on the right, and the other one, the blue one, is also going to be present. And they're going to be used as templates to make a new cell. I'm sorry, to make the complementary other chromosome. And I'm going to just draw this other chromosome in a different color. Let's go with green. So each of these are used as a template, and then it's used to make a new chromosome. So it's important because each one of these chromosomes has all the genetic information in it to copy and make another complementary chromosome, so that in each resulting cell, we preserve half of the original chromosomes, the red one and the blue one. And it's used as a template to make the next one. Conservative replication, right? We'll still draw the different cells here. We'll still start with red and blue here. I think this will work out fine. And with conservative replication, when you produce a new cell, as it's copying the DNA, the entire original chromosome is conserved. So you're going to keep the same ones here. And they're still going to be bound to each other. And somehow, these chromosomes that are together are used as a template to make a new one. And I'll use the same green just to keep the pattern the same. And so it'll make this here. It'll make a brand new copy of the chromosomes. Now, conservative replication doesn't happen. This is just an idea, because when they did the experiment, they didn't know. So they had to think about all the different kinds of ways replication could happen. And one was semi-conservative, one was conservative, and the other is dispersal. So that's why it's significant, because it preserves half of the chromosome in each generation. And then with conservative, I mean semi-conservative, if you think of the Nielsen and Stahl experiments, I'm going to have to redraw these here to make them make sense here. Originally, they grew them in N15. So all the DNA was made in media containing N15. And so here I'm just going to put blue as N15. And red I'm going to put as N14, the lighter isotope. So they grew them in N15 DNA. I'm sorry, they grew it in media containing the heavy isotope N15, nitrogen 15, so that the DNA was heavier. And so when you ran this on a cesium chloride gradient, the DNA from here, it's going to be at this lower level here that you would predict heavy DNA to move to when you spin it. Remember, 14 is up here, would be up here, like so. So with semi-conservative, remember, that means half of this is going to be conserved in each of these cells, of the new cells, right? And the new DNA that's made is going to be used with N14, because remember, we get rid of all the N15 from the media, and we add in only N14. Meaning all the new DNA is going to be lighter. And with semi-conservative, that heavy one, the blue one there, it's used as a template to make new DNA, and that new DNA only has this light isotope N14 to make the DNA. So it's going to be lighter, and it's going to be the same for both of these. So now when you run this on the cesium chloride gradient, after one generation, all the DNA is half heavy and half light. So you would predict that that DNA would be somewhere between N14 and N15, which I'm trying to show here. And now if each of these cells divides again, we'll have 4 cells. Horribly drawn, but you get the point. So this cell here, when it divides, both those chromosomes are going to split apart. And they're each going to be used as a template. So we'll put that in here. So this cell here, when it divides, it makes these 2 cells. And one of them is going to use the red one as a template, and the other will use the blue as the template. Same thing here, when this cell divides, it's going to use the blue as a template for one of the new cells, and it's going to use red as the template for the other new cell. But when it makes the new DNA, again, we still only have N14 in there. So all the new DNA is going to be of the N14 variety, the lighter isotope. Shown here. And when you run this on the cesium chloride gradient, you'd expect half of them, because half of the cells only have N14 DNA here and here. So you expect about half of them to be at this lighter isotope up here where you expect N14 to be. But you also still have the hybrid, half heavy, half light. And so that's going to still be in the middle. And nothing is going to be at N15 anymore. That's gone forever. So this is generation 1, this is generation 2, generation 0. So every time it divides, it's going to cut this in half. And because of that, if you move to generation 50, way down the line, the same phenomenon is going to be observed, because every time these split, it's going to use each half as a template. And so you're going to continue to get more and more DNA at this lighter variety N14. And you're still going to get a minor portion here, but it's going to make up a very small amount of the total amount of DNA present. It's still going to be there, but it's going to be minor. Okay. I'm not sure if you want me to say more about that. I'm happy to. I notice I want to check something here. Okay. All right, so let's see if there are other. Could you go over application question E from day 6? Maybe. Let me see what that question is. All right, so this one I cannot go over because I said at the end of class today and Monday that this question is going to be part of the group exam. One that you could, the first 10 minutes of the exam, you're going to answer 2 questions as part with your team. And this is one of them because we didn't get to class. So I said, I'll make this part of the group. So you don't have to worry about what those questions are going to be. I'll come back to this in a moment so I can talk a little bit about it, but I can't give you an answer. Same with this one. I said this question also will be on the group exam. So I can't go over this in any detail because it's going to be part of the exam. But I can tell you that what you're going to have to do is decide which of these 3 types of horizontal gene transfer methods is most likely responsible for Allen's new infection. One thing I want to make sure is clear is that we talked about these 3 different types of horizontal gene transfer mechanisms: transformation, transduction, and conjugation. Only of those 3, only transformation can occur between different species. Transduction, you can only transfer DNA from one species to the same species. Conjugation, you can only transfer DNA from one species to the same species. Transformation, those bacteria don't care. They'll take up DNA from any source. They'll take up other bacterial species DNA. They'll take up human DNA if necessary. They might not be able to do anything with human DNA, but they'll be able to take it up, right? So they can take up any DNA. And so just make sure you understand the distinction between those, that one distinction between the 3 that I'm not sure if it was real clear in the podcast, but we talked about it in class, but just make sure you know that. This one here, we talked a little bit about the HIV in the podcast, so make sure you go back over that. And I'm telling you here that this drug, AZT, is a base analog that binds to and blocks the function of the enzyme reverse transcriptase. Then you have to think about the processes that HIV uses in replicating itself and decide which one of these 4 is it going to most likely impact. HIV's ability to diffuse with outside of cell, conversion of HIV's RNA genome, integrating the HIV genome, or forming new HIV particles. Then I want you to address this question here, that usually when somebody's treated with HIV, when patients are treated for HIV, they don't just give them AZT or just one of the drugs that goes after one of these 4 or more processes, but rather they give them a cocktail of 7 to 10 different drugs that goes after different ones of these features of the HIV. I want you to think about, like, why is it necessary for someone who has HIV to get this cocktail of 7 to 10 different drugs as opposed to just one drug that attacks just one thing? Okay. So I hope that helps, but I think that's as much as I can tell you, since this will be an exam question that you'll have as a group. Any other questions about concepts that you're unsure of? Application questions? The exam? see someone typing here, so I'll wait a moment. So the exam, as I said in the beginning, it'll have between 30 and 35 questions. It'll have between 25, 28 multiple choice questions. It'll have three or four short answer questions, probably closer to four. And it'll have 3, 4 matching questions. So more multiple choice questions, but there are also lots of at least 4 short answer questions. And one thing, when you think of the application questions that you've done during class, don't think of those as questions that could only be short answer questions on the exam. Those application questions, I could, and I will, write multiple choice questions that use the information or ask you to analyze things from like a short answer, an application question that you had in class. So sometimes students will hear four short answer questions and they're like, oh, there's only 4 questions on the application questions. Don't make that mistake. There's more than 4 questions there are more than 4 questions on the exam that will ask you to apply your knowledge in a way similar to the application questions. In this syllabus, it says that all exams in the final are 55%. What are they individually? 11%, because there's 5 of them, so they're all equal. I know a lot of times when, like, the first exam you take in a class is always the most nerve-wracking because you don't know the style and or the format or what to expect. And so the best I can tell you is that study the note-taking guides and the application questions. Focus on that. If you have, by chance, happen to have the quiz questions we ask at the first part of every class, you can look at those, but I don't necessarily think those are the best way to spend your time as you're studying. There will be some questions that are that kind of level, like recall questions. But if that's all you study, or you spend the majority of the time studying those short answer questions, it's probably going to not be a good result. So make sure you study intensely the note-taking guides and then the application questions. I think the homework problems are useful and you can go back and look at those, but when I write the exam, I don't look at the homework questions. Those are just questions to help cement some of the core knowledge. So they could be helpful to look at, but probably not the most helpful compared to application questions and the note-taking guides. See, one more question maybe coming in. Feel free to put a question in the chat or you can just ask it out loud. That is fine too. Maybe there wasn't a question. I thought there was a question coming through. I mentioned some questions. I said I guarantee you that they're going to be on the exam, or a variation of that question, right? Make sure you know those things, and we can kind of just skim through these, I think. Things like drawing. The only way I could make you draw something is on the group exam, because the other exam is on your laptop, so that won't work really well. So I'm not going to ask you to draw anything. I already told you the 2 questions on the group portion. It's useful to be able to draw things because it helps you learn it, but I would not waste your time making sure you can draw a double-stranded helix in its entirety. What you should do, though, is make sure you can look at examples of a double helix and point out things that are wrong, or be able to identify different parts of that double helix. Now, that would be useful, and I would almost guarantee a question like that will be on the exam, where you have to look at the double helix and make some predictions about it. I always ask a question like this, guaranteed. Sometimes it's a short answer question, sometimes it's a multiple choice, but probably by and large, it's usually some version of a short answer kind of a question. It won't be the same sequence. It won't be these exact same questions, but you should be able to look at a piece of DNA sequence and tell me why, and be able to answer questions about the number of hydrogen bonds, the number of deoxyribose sugars, ribose sugars, purines, phosphodiester bonds. You should definitely know this. And you should also know this general concept here, of course, about why one piece of DNA might be more difficult to pull apart because it has more GC bonds. Let's see here. You should know the Hershey-Chase experiment, the Muselson-Stahl experiment, and the Griffiths experiment as it relates to knowing and discovering what the genetic material was. We spent a lot of time talking about the Hershey-Chase. And so I would be surprised if there wasn't a question about Hershey-Chase. But if I were to ask you this question, and I'm not saying I am, this isn't a guaranteed question like I said with the other one, but to talk in detail about the genetic material and the experiment about the genetic material, I might focus more on Muselson and Stahl, since we talked about Hershey-Chase in class. But again, that's not one of my guarantees. It's a good idea to think about all of those experiments: Hershey-Chase, Muselson and Stahl, Griffiths, and what would those results have looked like if protein was the genetic material, or if RNA was the genetic material. I know in the past I've asked a question about, like, if proteins were the genetic material, what would the results of these different experiments have been? It might be a multiple choice, it might be a short matching or something like that. But to know what each of these groups of people who did these experiments, what they would have seen if proteins were the genetic material, I think that's probably pretty important to know. I'm skipping through some of these, and that doesn't mean I don't think they're important. I'm just trying to think of some things that kind of highlight as I'm going through. So this is a good experiment because it talks about why we know that humans' genetic material is also DNA. You should be able to look at examples of cesium chloride gradients and tell me if it's semi-conservative, conservative, or dispersive. And again, I'm not going to make you draw any of this because I really can't on this based on the style of the exam. You can have scrap paper with you so that you can draw things out. That would be fine. Again, I'm not going to make you draw the replication fork, but I think it's really a good idea to be able to look at a fork like this. And by looking at the fork, be able to identify the different proteins and enzymes that are present on it. So if you had a picture and I showed you like where helicase was, where gyrase was, right, something like this, you should be able to point things out. You should also be able to identify where the 5' and 3' ends are. Usually when I draw this, I go 5' on top to 3' and 3' on bottom to 5', but that might not be the case. What if I showed you a strand like this, or a fork like this, and didn't tell you where the 5' or 3' ends are, but asked you to identify what is this one? Is this one 5' and 3'? What is this one, 5' or 3'? And same here. In fact, let's do that. Look at this. On your own, tell me where the 5' and 3' ends are for all of these. I'm going to pause for about 2 minutes so you can take a peek at this. Keeping in mind where you see the Okazaki fragments coming off here, and where you see the leading strand moving into the fork. Okay. This first one here, or if we do this first arrow right here, is that 5' or 3'? You can put it in the chat or you can just yell it out. Does anybody want to guess? As you're looking at this, remember, get this, well, don't actually get this tattooed on your arm, but think of it as a tattoo, right? That DNA is red. Well, somebody, you tell me, how is DNA red? Is it red 5' to 3' or 3' to 5'? It is red 3' to 5', and thank you, Addison. And new DNA is laid down 5' to 3'. So when you're looking at this, we know that DNA is going in this direction, right? So if it's moving in this direction, it's reading the DNA in this down. Well, somebody tell me real quick, because I want to make sure. Can you see my cursor here, like going all around crazy like? Okay, I want to make sure if I'm pointing to something, you know what I'm pointing to. Okay, good. All right, so we know that since DNA replication on this bottom strand is moving from right to left, it's got to be reading the DNA this way. And if it's reading the DNA this way, that means this end here, where this arrow is pointing, that's the 3' end. And if that's the 3' end, then that means this one has to be the 5' end. And once you identify one of these, the rest just falls into place, right? Because if that's this is the 3' end, and that's the 5' end. If it's anti-parallel, then that means this one here has to be 3'. And that means this one here has to be 5'. And that means that this strand here going, say, here, right? The new DNA is 5' here and 3' here. And 5', all of these fragments are going to be 5' to 3'. Same here, because it's got to be anti-parallel. So this one's going to be 5'. This one's going to be 3' of the new DNA, right? Because it's always going to be synthesized in the 5' to 3' direction. It's always read 3' to 5'. So be familiar if it looks like this, or if I gave you a fork that looked like this, where it's 5' to 3'. Because the way the leading and the lagging strands are will be different. But make sure if you see a fork, you can label all the parts on it. I almost always ask a question like that. And be familiar with when you like draw a fork here with everything on it, think about what each of those enzymes does, what they each do, and then what would happen if they didn't do their job. And so it's a good way to kind of memorize and learn about what each of them are doing. We talked about the gyrase inhibitors, thinking about how you can take advantage of the differences in the way eukaryotes and bacteria replicate their DNA. If there's any differences, you could exploit that to make an antibiotic. Make sure you understand the different enzymes and the different DNA polymerase enzymes in eukaryotes and what specifically do they do. I could give a scenario where you have to think about what might happen if those enzymes weren't working, right? So, we talked about xeroderma pigmentosum, and when that wasn't working right, we linked that to the DNA polymerase ETA. And so think about things like that, or if it's the main one, what would happen if the main one wasn't working right? What would happen if the mitochondrial DNA polymerase wasn't working right? We already talked about telomeres. Just really be familiar with the fact that telomeres they determine the life of the cell, and as long as the telomerase is there, those cells will remain immortal. They'll keep dividing at the same rate and they won't die. But when telomerase isn't there, then those cells, when they divide, they have a finite life. I'd be shocked if I did not have a question about a PCR program where you have to determine what would happen if a particular step was changed. Okay, 5 here. So same thing, if you were to look at chromosomes, you should be able to identify the different parts of a chromosome, where they would be. Really make sure you understand heterochromatin and euchromatin and what their differences are, where you might see heterochromatin, where you might see euchromatin. I'll probably ask a question similar to this. And as I said, like in class, we talked about how different genes in different cell types, the same gene was present multiple cell types, but it's only expressed in one cell type. And so be able to distinguish between those. I also said on your own thing about like if this was not a neuron, but if it was a, I'm just gonna put E for embryonic cell. And this one here was a pancreatic cell. I'm sorry, an adult cell. This was an adult cell. And an embryonic cell and an adult cell each have the same genes in it, but maybe we're going to look at an embryonic specific cell, like embryonic hemoglobin. If we're looking at embryonic hemoglobin, which one of these is going to be expressed? Well, it's going to be expressed in the embryo because it's an embryonic form of hemoglobin. And if it's expressed in the embryo, that means in the adult, the nucleosomes are going to be attached to prevent it from being expressed. I could also have the same question, but now I'm looking at adult hemoglobin, then it's just going to be reversed. Right? So the same concept of the way we regulate which genes are turned on in which cell and in which developmental stage we're at. I won't ask you anything specific about talking about how much DNA we have in our body, but you should be aware that we have so much DNA in our body and that the ability to pack it and unpack it in precise ways to turn on necessary genes, you should be aware of that. Understand kind of the critical importance of how when DNA is packaged up into chromosomes using nucleosomes, that it protects the cell from any enzyme that might be able to get to it, a transcription factor, a replication factor, any enzymes, a DNA, because as long as those nucleosomes are attached to the DNA, it will protect it. And then this is from today, or yesterday, depending on which class you're in. And so this is more just making sure you're aware of how to, what do some of these terminologies mean? Like what this means it's a auxotroph and it cannot make its own methionine, biotin, and then it can make threonine, leucine, thymine. Make sure you're familiar with those kinds of terminologies. In this particular one, make sure you understand that the importance of having multiple mutations because that prevents reversions of the mutants to pluses or wild type, the natural version. And so why that's important and what these results would have looked like. These results would have looked like if there were reversions, right, we would have seen colonies on these plates, but we didn't. And since we didn't see colonies on the left or right, we know that the colonies in the middle are very likely not because of reversions. They're more likely because that there was some exchange of genetic material between two different cells. I said I would ask a question about this, and I would be shocked if I did not ask a question about this. It'll be similar, but it's not going to be the exact same, but it'll be similar. And as I said in my class today, it could be worded in such a way where there's multiple correct answers, that the resulting cell could grow on multiple medias. If that's the case, if I ask a question like that, I won't say pick all the correct answers. I will tell you how many of them are correct. So if there are 2 or 3 correct answers, I will tell you that pick the 2 or 3 correct answers. Or two, whichever it is. I won't be vague. It'll be 2. If there are 2 answers, then I'll say there are 2 choices, pick both of them. We talked today about, and yesterday, about how to design this so we know it's transduction and not transformation or conjugation. And I said, on your own, do this, right? How could you do that to show its transformation? So you can think about how you would set that up in that way. And then we did not get to D and E, and I told you before that those will be the 2 questions on the group exam. Okay. All right, I don't know if you have questions, still have questions, but I'm happy to answer other questions you might have. And if there's nothing else, I'll probably end things in about a minute, but I can stay here for a little bit longer if you have questions. I'm witnessing the mass exodus. And so I'm going to stop sharing, and then I'm going to turn off, I'm going to stop recording.